Translation is the actual building of the polypeptide chain from RNA. There are three important components of translation, or three molecules that you need to be familiar with. mRNA is messenger RNA, which we learned in transcription is made from the DNA template. The second molecule that you should know is tRNA, or transfer RNA. This is the interpreter or the translator of the mRNA strand. And also the ribosome, where the actual translation is taking place. And please note that translation is the building of that polypeptide or protein. This diagram is something that you should study and understand. There's a few important things that are on this diagram or picture. You can see on the left the brown blobs. That's the two parts of the ribosome. On the bottom is the small unit, and on the top with the EPA is the large unit of the ribosome. Also, you can see there are three sites in the ribosome: the A site, the P site, and the E site. When translation takes place, it works along the mRNA strand from the 5' cap to the end where the poly A tail is added at 3'. Something else that you should notice on here is the tRNA, which in this picture it looks like a Christmas stocking upside down. The Christmas stocking, or tRNA molecule, is what brings in the appropriate amino acid. Now, how does it know what amino acid to bring in? It also has a code on it as well. Its code is called an anticodon. The anticodon is going to match up or complement the codon in that mRNA strand. And when they match up, again with base pairing rules, that signals that the appropriate amino acid is made and it can be released into that polypeptide chain in order to build the protein. You should understand a little bit about tRNA since it's so important in translation. First, it is transcribed or synthesized in the nucleus. Every tRNA molecule is specific for an amino acid, which is transferred to the ribosomes, and the tRNA molecule also has a code on it, a 3-letter code or nucleotide triplet. This is known as an anticodon. The anticodon is going to complement or base pair with the mRNA strand codon. When these complement one another, that indicates that the appropriate amino acid has been brought and can be added to the polypeptide chain. How does the right amino acid attach to that tRNA molecule? It is by an enzyme called aminoacyl tRNA synthase. And you don't have to know the details about this, but you should know the name and what it does. The tRNA molecule needs an enzyme in order to bind to the right amino acid in order for translation to occur. Ribosomes are the site of translation or building a protein or a polypeptide chain. So what exactly is a ribosome? So a ribosome is made of rRNA or ribosomal RNA, and it's also made of protein. It is synthesized in the nucleolus, and there are 2 subunits to it. So if you glance at the picture, again, usually proteins, enzymes are drawn as blobs. Usually we're used to seeing ribosomes drawn as little circles, which in fact is incorrect. there's two units on the right. You can see the cream-ish color. That's going to be the small subunit on the bottom. The larger part on top, which is light blue, is the large subunit. A little bit more about the structure of ribosomes. There are 3 active sites, 3 places that contribute to the building of a polypeptide chain. The first site is known as the A site. This site is going to hold the tRNA molecule when it brings in an appropriate amino acid. The P site is going to hold the growing polypeptide chain. The E site is where the tRNA is going to exit after it has deposited its appropriate amino acid for the chain. Similar to transcription having 3 steps, translation also has 3 steps. The first step is known as initiation, and the beginning of initiation, again, building a polypeptide. It begins with the transfer RNA molecule attaching to the P site on the ribosome. That's the very first step that is initiated. After the tRNA is attached, and it has to have methionine attached to it, since that is the first amino acid in a polypeptide chain. Once that's attached to the ribosome, then the mRNA is going to attach. It has to start with that 5' cap, and then the ribosome essentially reads the mRNA for the start codon. And finally, the last step of initiation is bringing in that large subunit to the top and putting it all together for building of a polypeptide. The second step I want to show you in a few different slides so you can look at the diagram and see what's changing between each slide. The second step of translation is called elongation, which is making the polypeptide longer and essentially building the protein. The first part of elongation is codon recognition. This is where the tRNA anticodon needs to match the codon in the A site. So when you see the ribosome from right to left, reading backwards, the site is going to be A, P, E. A being on the right, P being in the middle, and E being on the left. So again, codon recognition, tRNA anticodon needs to match the codon in the A site first in elongation. And please remember that the P site is already filled by the tRNA with methionine. Something else that occurs in the second step of translation, again called elongation, is the actual peptide bond formation. So that's the type of bond that keeps amino acids together in a protein or in a polypeptide chain. So if we look at this diagram, on the second step where it says peptide bond formation on the right, you can see that the existing polypeptide chain is getting transferred to the right to the A site to make that polypeptide longer. Now, basically what's going to take place is that ribosome is going to move towards the right. That's going to shift the tRNA molecules into a new position. the process of the tRNA molecules being shifted into a new site within that ribosome is called translocation, which is the final part of elongation. So again, please really look at this diagram and see how the tRNA molecules are moving within the ribosome. So if you look at the ribosome where it says number 3, translocation, the ribosome, the tRNA molecule that's in the E site is exiting. Again, it is already given its amino acid to the chain. The ribosome has shifted towards the right or towards the 3' end of mRNA. The polypeptide is building. And again, this is occurring at a pretty fast pace, building this protein. The last step in translation is known as termination. In termination, the stop codon is reached and translation is signaled to stop. Now, how exactly does it stop, and how does the polypeptide get released from the tRNA and the ribosome? A release factor is going to bind and signal that the polypeptide needs to be released. If you look at the diagram, you can see a release factor that's being added to the A position or A site of that ribosome. It's going to attach to the stop codon in that mRNA strand. Now, once that happens, another tRNA molecule cannot come in to bind and build and deposit its amino acid to that growing chain. Once that takes place, the tRNA molecule is released, the polypeptide chain is released, and the two subunits of that ribosome are going to separate or dissociate from one another. So there are many things that happen to these proteins or these polypeptides once they are assembled. So just mentioning a few, sugars, lipids, phosphate groups can get attached to these polypeptide chains. Enzymes can remove amino acids from the ends. The polypeptide can get cut into several pieces. So sometimes a polypeptide in its long version, it isn't effective, but when it gets cut into several pieces and makes bonds with one another, then it's active in a sense. And insulin is actually an example of that. And also different subunits can come together. So hemoglobin is an example of that. So you have different polypeptide chains together on their own. They can't operate in their function of moving oxygen through the bloodstream, but when all four are put together, then it can do its job. To recap from one of the old chapters, there are two kinds of ribosomes, and they do different jobs based on where they are. They both make proteins, but they make certain kinds of proteins depending on where they are. Free ribosomes, they synthesize proteins that stay in the cytosol and that are going to function there. Bound ribosomes, again, bound to the endoplasmic reticulum, these make proteins that are of the endomembrane system, the nuclear envelope, the ER, Golgi apparatus, lysosomes, vacuoles, plasma membrane, and for proteins that need to be secreted. Now, how